Which of these RNA types brings amino acids to the matching mRNA codons? Is it rRNA, mRNA, or tRNA? Yep. tRNA. Which of these RNA types makes up the ribosome? rRNA, ribosomal RNA. Which of these RNA types carries the message that was produced in transcription? mRNA. And we don't want to reveal. We'll skip that. Okay. Oop, you ready? Sorry, I'm going too quick. That was easy, right? Message. Ribosome. I don't know. There's no one to say that. Okay. Protein synthesis. Summarize the events of protein synthesis. What happens first? How do I do protein synthesis? What do I do? I go from. What do I start with? What molecule is this? What is this? What molecule is that? What is it? Trey, what are you thinking? DNA. I go from DNA to what? RNA. And what's that process called? Transcription. And I go from RNA to. What's RNA turn into? mRNA, what does it turn into? Protein. Translation. What's produced in transcription? What do you get at the end of transcription? So transcription, I get. RNA. Translation. Protein. Amino acids. Which one uses DNA? Transcription, translation. Transcription. rRNA. Easy, right? Go up to the other page. Is everyone good here? Yeah. tRNA, rRNA, mRNA. We are starting today on page 63, your last section. Three. What do I want? Unit 6 will be your last unit we'll do together. I encourage you to come after school tomorrow to do the other units if you so choose. Dang. Come for like an hour? Can you come 2 to 3? Oh, it's okay. It's okay. You can also come on Sunday's last minute questions if you have any. It's okay. Sunday at like 7. That'll be questions and answers. Tell me, what is heredity? Genes, what about genes? What am I doing with my genes? Huh? Stuff that runs the family. So heredity, I am doing what with the genes? Passing them. It is the passage of traits from parent to offspring. What is an allele? An allele. It is the form of what? Form of a. What are we talking about? It is a letter. It's a form of a gene. Hi, letter, like A. When I say I'm a dominant allele, that means I am what? Uppercase one. So I'm always expressed. So the recessive must be the lowercase letter. So this one gets covered by that dominant allele. So you need two of them to express it. What is a genotype? Or give me an example of it. My genotype. My genotype for hair color would be yeah. Give me an example. Uppercase A, lowercase a, right? It is your combo of alleles. Your genetic makeup. What is phenotype? So if genotype is my genetic makeup, what is phenotype? What did you say earlier? Physical. Maybe you're purple. Maybe you're tall. What does it mean to be homozygous? What does homo mean? Same. This is the same alleles. Otherwise known as purebred. Capital, capital, lowercase, lowercase. Homozygous recessive, homozygous dominant. What about hetero? Hetero means different. Or a hybrid. For each genotype, tell me, is it hetero or homozygous? Here, let me zoom in a little bit more. Capital A, homo or hetero? Homo. Capital B, lowercase b. Capital D, lowercase d. Capital E, lowercase e. hetero. What about my G's? N's? R's? Q's? T's? Good. So if I said short hair is dominant to curly, what would be my genotype? What would be my genotype if I said I had straight hair? Give me a letter. Big letter. S. Okay, so what would it be if I had straight hair? What letter combos would I have? Uppercase S, lowercase s, right? What else can I have? That's straight hair. Two uppercase, right? And if I had curly hair, I would have two lowercases. If I said brown eyes are dominant to blue eyes, capital B would give me what color? Brown. Capital B lowercase b would give me. And two lowercase would give me blue. Easy, right? Remember that? This one was easy. Okay. This is the easy one. So on to our Punnett squares. Monohypercrosses is when I have one trait and the dominant overpowers everything else. So dominant's always gonna win. So in a green monster, having three tails is recessive to having two tails. Create a homozygous two-tailed monster with a heterozygous two-tailed monster. So what letter should I use, do you think? What letter do you want to use? If I'm talking about tails, I'm probably going to use T. So capital T is two tails or three tails? Which one's dominant? Two. I'm crossing a homozygous two-tailed monster. So what is their genotype? If I'm homo, two. What do I look like? What's my genetic makeup? Homo means same, and two is what letter? Capital T. Okay. And I'm crossing it with a hetero two, so what's their genotype? Do you remember how to solve it? Drag it down, drag it over. So I get two of them, homodominant. Two of them, hetero. Zero, homorecessive. Phenotypically, all four of them will be two tails. And none of them will have three tails. That's monohybrid. Incomplete dominance is when my dominant isn't strong. So they blend. Red plus white equals pink. In radishes, red and white are purebreeding colors. Hybrids get purple. If red is crossed with a white radish, what is the phenotype of the F2 generation, assuming the F1 generation self-pollinates? So I'm crossing R for red, lowercase r for white. My parents are this. F2. It asks me for F2 because we're going to self-pollinate. So I set it up. Solve it. I get a 1, 2 to 1. So 1 is homodominant, 2 are hetero, 1 is homorecessive, 1 is red. Purple is my intermediate. That's 2. Ooh, it's next. No, it is right. Okay, 1. And I wind up like that. The same way you would do this, the Punnett square the same way, but knowing that the dominant isn't strong, I start blending. Whenever it tells me my hybrids, my heteros are different colors, is an example of incomplete dominance. Then you're on the back. Ready. I don't need more time. Okay. Codominance. Move this over. Co means what? Together. So they're both dominant. So I wind up being spotted. A new species of carnivorous cats have been called the gats. In cats, black and white spots are codominant. If a heterozygous spotted gat is crossed with a white spotted gat, what's my phenotypes and genotypes? So I'm either black, big B, or white. I am crossing a white spotted gat, so I'm WW. What do I get? Set it up, drag them down, drag them over. 50. So 0 are homo for B. 2 are hetero. Ooh, 2. Messed up. It's 2. 2 are homo for W. So 0 black. Spotted 2. White. 2. Monohybrids. Multiple alleles. Multiple alleles. Morning. Multiple alleles are when the babies think are typically referring to blood types. So our example here, we think our baby was switched at the hospital. DNA fingerprinting technology does not exist yet. Mom is O, father is A, B, and the baby is B. Is this their baby? So mom is type O. Remember, O is recessive to A and B. So mom is little o, little o. Dad is AB, so he's capital A, capital B. And the baby's type B, so he can either be homodominant or homorecessive. So was the baby switched? We crossed dad with mom. Was their baby switched? Yes or no? Is this their baby? Yeah, it's their baby. They had a 50% chance to be BO. So it's their baby. And then your last one is dye hybrids. Dye hybrids are going to be a little bit more intense. Remember, they are where we do 2 trait crosses. Some fruit flies' red eyes are dominant to black eyes, and long wings are dominant to short wings. Assume that a cross was made between the fruit flies of the genotype capital A, lowercase b, and with those of genotype capital lowercase a, capital B. Give the Punnett square of the F2 progeny. Hint, F1 produces F2. So I know what F1 is. It's right here. So F2, since they can only give one of each, is going to be this person. Because it's the only possible baby. I would have to FOIL it out. Remember, that's where you go first. So I get capital A, capital B. Outer 2, capital A, lowercase b. Inner 2, lowercase a, capital B. And last, lowercase a, lowercase b. They're going to cross with each other, so I line them up. And I solve it. Remember, I have to do that FOIL step because I'm making a gamete. They can either give me the capital A or the lowercase a. They can either give me the capital B and the lowercase b. They're not linked, so they can give me either or. So drag it down, drag it over. I'm going to drag all my capital A's down. Drag them over. Then do the B's. Solving it out. And then I check to be red with long wings. I know red when I look up here is capital A. To have long wings are dominant, so they are going to be B. So how many have a capital A, capital B? I go and count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. Red eyes with short wings. I know red eyes are capital A's. Short wings are recessive, so I can only be lowercase b. I go and count them. That's 1, 2, I think it's 3. 3. Black eyes are recessive, so they're going to be lowercase. Long wings are still dominant. So I go and count them, and I get 1, 2, 3. And all recessives right here is 1. And then your last section, your sources of genetic diversity. Why do I care? I try to figure this all the way out. sources of genetic diversity. Remember, we want to increase genetic diversity because it leads to better survival. The more diverse you are, the more skills you have, the more chances of your survival if something was to happen. And you're going to reproduce better. You're going to be able to pass on your traits, have some babies, and live your best life. The way we do that, we have our lovely crossing over. That's where the X's line up across each other. So we're exchanging material. You'll have independent assortment. Man, why is still a door locked? Thanks. Thanks. Basically, how do the cells line up? Here. The independent assortment is which chromosome lines up which side, determining which cell they wind up in. Both of those are going to give you a unique gamete, meaning you're going to be different from your siblings. Sexual reproduction increases genetic diversity because which gamete winds up winning, there's a chance that any sperm can fertilize the egg. Who wins the race, basically. And then mutations always change the DNA and they introduce new alleles. And that's the end for Unit 6. So what you doing today? Your last set of stations. Task 1 is vocab. Task 2 is genotype to phenotype. Task 3 is monohybrid. Task 4 is dihybrid. Station 2 is incomplete and codominant vocab, compounded squares. Task 2 is about blood types and multiple alleles, so they work the same way as normal ones. And station 3 is multiple choice questions, multiple choice. And then you have your quiz on page 68. You're going to page 68. It's your last section to do. So at this point, you should have up to page 68 done. That is what you get graded on. You will see Unit 7, Unit 8, and Unit 9 have their own recap sheets. Those are what's going on after school tomorrow at 2 o'clock. So you're basically done with this book.